This is Bernard Paulson. I will now speak to the material in chapter 13 in this book. This is the first chapter in part 4 of the book, which is the last part. In this portion of the text, we will learn about macromolecular systems and how to describe them in the context of simulators for low molecular weight metabolites. Macromolecules, like protein, have typically many ligands binding to them, and depending on the ligand-bound state of the molecule, it will have different functions. So the mass action effects associated with the different ligands control the activity state of the molecule. In this lecture, we will look at the function of hemoglobin and how it is regulated in the context of the human red blood cell. I will talk about the role of red blood cell glycolysis and the production of a key intermediate in red cell glycolysis that binds to hemoglobin and thereby regulates its activity. We will talk about how one builds a subnetwork module around hemoglobin and its bound states. Once we define the environment for this subnetwork, we can compute its steady state, and in fact we can then simulate the deviations from that steady state in that module by itself. What we can then do is to integrate the glycolytic simulator from chapter 10 with this hemoglobin module to build an integrated model that describes the response of hemoglobin to oxygen and how the oxygen-bound state of hemoglobin interacts with glycolysis as a pathway. First, we'll talk a little bit about hemoglobin. We all know that hemoglobin is a heterotetramer comprised of 2 alpha units and 2 beta units, alpha globins and beta globins. In each one of these monomers, we have a heme molecule that is indicated here with a red circle that chelates an ion atom. That ion atom has to be in the +2 charge state in order to bind to oxygen. If it is oxidized to the +3 state, it is non-functional. Hemoglobin in red cells is neither synthesized nor degraded, and therefore is present in a constant total amount. The state of hemoglobin is influenced by oxygen concentration that in turn is equilibrated with the concentration of oxygen in plasma, and it is also influenced by an intermediate called 2,3-DPG, 2,3-diphosphate glyceride. In this diagram, it is designated as DPG-23, synonyms. The hemoglobin molecule has 4 binding sites for oxygen, and the 4 equations, chemical binding equations, shown at the bottom of this slide, show the sequential loading of hemoglobin with oxygen. 2,3-DPG is a unique metabolite to the red blood cell, and it is made in a side branch or a shunt to glycolysis in the red cell as indicated. 2,3-DPG is made by DPG mutase that takes the phosphate in the 1 position in the natural glycolytic metabolite 1,3-DPG and moves it to the 2nd position. The concentration of 2,3-DPG in red cells is very large. It's in the multi-millimolar range and is almost equimolar with hemoglobin, which is also in the multi-millimolar range in red cells. 2,3-DPG will bind to hemoglobin and convert it to what's called a deoxyhemoglobin state. This is a T state or a tight state of hemoglobin that cannot bind to oxygen. So the higher the concentration of DPG, the more of hemoglobin is converted to the deoxyhemoglobin form. 2,3-DPG is therefore a key regulator of the state of hemoglobin by manipulating or regulating the amount of hemoglobin available for oxygen binding. Most of us are familiar with this curve here, this S-shaped curve that shows the oxygen saturation of the hemoglobin molecule as a function of the partial pressure of oxygen. Now, 1 atmosphere is 760 millimeters mercury, and 20% of that would be 152 millimeters of mercury. So that's the partial pressure of oxygen in air. Because oxygen has to diffuse across the epithelial layer in the lung, the partial pressure of oxygen in the bloodstream in the lung is closer to 100 to 120 millimeters of mercury, as indicated. In tissues, on the other hand, the partial pressure of oxygen is much lower, closer to being in the 30 millimeter mercury range, plus minus 5 or so. Therefore, the concentration differential between the lungs and the tissues of saturated hemoglobin with oxygen gives the amount of oxygen that is released from hemoglobin as it travels through tissues. And as you see, when there's no 2,3-DPG present, the curve shifts to the left, and if DPG concentration increases, the curve shifts to the right. This is the way 2,3-DPG regulates oxygen delivery to tissues by controlling the saturation curve of hemoglobin with oxygen. Hemoglobin, we should note, increases the oxygen content of blood by almost 2 orders of magnitude over aqueous solution. Oxygen is very sparingly soluble in water at about 7 ppms at 37 degrees, where ppms is part per million. In fact, it is this elevated oxygen content of blood that allows for the high volumetric oxygen consumption rates in tissue that in turn basically enables animals to be mobile and do a number of things that they couldn't do without that metabolic intensity. Here is the shunt that makes 2,3-DPG on glycolysis. There is the mutase, DPG mutase, that moves the phosphate bond from the 1st to the 2nd position. And then there is the DPGase that splits off that phosphate in the 2nd position generating the glycolytic intermediate 3-phosphoglycerate. 2,3-DPG, as shown to the right, can bind to hemoglobin and move it into the deoxy state, and that is an equilibrium, a rapidly equilibrating reaction, as shown at the bottom of this slide. So these are the ligands then that control hemoglobin, 2,3-DPG and oxygen. There are others which we will not consider here. Now we need to characterize the state of hemoglobin, and we are going to do that by forming pools and ratios as we were introduced to in earlier chapters. We're going to define the oxygenated or oxyhemoglobin state, the oxygen-bound state of hemoglobin, that we're going to call oxyhemoglobin, as shown in the top equation, where we add hemoglobin with one molecule plus two hemoglobin with two oxygen molecules bound, and so forth. This simply counts the number of oxygen molecules bound to hemoglobin. If we ratio that to 4 times the total hemoglobin concentration, we get the loading of hemoglobin with oxygen. If this ratio is 1, all the hemoglobin is carrying 4 oxygen molecules. And then, of course, there is some fraction of that in tissues in particular. We can also characterize the state of the inhibitor 2,3-DPG by recognizing that there is a total pool of 2,3-DPG, either free or bound in the form of deoxyhemoglobin. We can ratio the bound state of deoxyhemoglobin to the total, and that gives us the fraction of 2,3-DPG that is bound to hemoglobin and forcing it into an inactive state or a non-binding state. We note here that hemoglobin total is a constant. Red cells don't synthesize or degrade hemoglobin, although it's probably deteriorating with age. I note that the red cell lives for 120 days, so any deterioration is much slower than the timescales we are concerned with here. And we note that the total amount of 2,3-DPG is a variable, as it is a glycolytic intermediate, as it's related to glycolysis, I should say, and can be synthesized and degraded. Okay, so that is the way we describe the state of hemoglobin, and we can use these ratios to get some measures of the physiologically relevant state of hemoglobin. What we will do now is to look at the steady state of this module and simulate its responses to the concentration of ligands. I would like to draw your attention to the image shown here, which comes from Goodsell's website, David Goodsell's website that we've encountered previously in these lectures. This shows the red cell membrane in purple, the plasma in green yellowish colors, showing a lot of albumin and other molecules. And inside the red cell, we see a lot of hemoglobin molecules shown in red. In fact, over 90% of the protein in red cells is in the form of hemoglobin. We will now set the steady state for this module. We note that oxygen will just come on and off hemoglobin in equilibrium relative to what is in the plasma, and deoxyhemoglobin is formed also in equilibrium or quasi-equilibrium relative to 2,3-DPG, as the ligand concentrations are changing very slowly. Here are the binding constants for the 4 oxygen loading steps shown. The numerical values for K1, 2, 3, and 4 are known. Here is the equilibrium for the binding of 2,3-DPG, and this binding constant is also known. This is the total mass balance on hemoglobin and all its states. We have hemoglobin 0 that has nothing on it, and then hemoglobin 1, 2, 3, 4 that carries oxygen, and then deoxyhemoglobin. We know the total. We can substitute in the equilibrium relationships given above into this mass balance to generate the equation at the bottom of this slide. We know the equilibrium constants, and if you know the concentrations of the ligands, oxygen and 2,3-DPG, as well as the total hemoglobin concentration, we can calculate the concentration of the free hemoglobin. And if we know that, we can go and substitute through all the equations above and calculate the concentrations of all the forms of hemoglobin. I am not going to detail this algebraically and would refer you to chapter 5, where we did the details of this type of mathematics for regulatory enzymes that have multiple ligands. We can now form the stoichiometric matrix for the hemoglobin subnetwork. Here are the concentrations that I've calculated for the different species of hemoglobin based on the concentrations of the ligands given in the top two rows. I should say in the last column, top two rows. The elemental balancing is shown here, and for all the internal reactions, they are fully elementally balanced, whereas the exchange reactions are not. For instance, we can see that the exchange flux for oxygen has two atoms of oxygen leaving or coming, depending on the sign of that flux. You will also note that the butase, the first column, puts elements in, and the second column for the DPGase removes the same elements, as both describe the input and output of 2,3-DPG, whose elemental composition does not change. I note that in the elemental composition portion of this composite table, that the hemoglobin is considered as an element because it never changes at all, and its elemental formula doesn't change in these reactions, and therefore this is a moiety that can be treated as an invariant. At the bottom, we have the numerical values for these equilibrium constants that I mentioned earlier, and we have in the second to last row the known steady state flux through the shunt that makes 2,3-DPG, which is about 0.44 millimoles per hour. Of course, all the binding reactions have a net flux of 0 because they are in an equilibrium state. So we have inputs here, inputs and outputs, which correspond to the ligands 2,3-DPG and to oxygen. We can simulate the binding states of hemoglobin in response to changes in the ligand concentrations. This slide shows the simulation in response to an oscillation in the oxygen plasma concentration, where the period has been set at 1 per minute, which is the average circulation time for blood, and the max and min are set based on the physiologically measured oxygen concentrations in the lung and in the tissue, and I mentioned these earlier in terms of millimeter mercury. Panel A here shows how oxygen is saturated with hemoglobin going through the cycle. It's very high in the lungs, which is the top of that oscillation, and low in the tissues when oxygen has been released. We can calculate the input and output of oxygen and see that it is positive when you're loading the red cell with oxygen, and it's negative when you're unloading it in tissues. So we can calculate the inputs and outputs there. We can, of course, integrate this over time to give us the net transport of oxygen into tissues. Panel C is interesting because it shows the dynamic state and how that is above and below the S-shaped curve we showed earlier. And this is, of course, the saturation curve for hemoglobin with oxygen. When we are above the curve, the oxygen charge of hemoglobin is above saturation, and oxygen can be released. When we're below it, we have the opposite case, and oxygen is being loaded onto hemoglobin. Interestingly, as oxygen is coming off and on hemoglobin, the concentration of hemoglobin without oxygen is going to go up and down, and that gives 2,3-DPG a chance to bind and form deoxyhemoglobin reversibly, and that reaction oscillates up and down as shown in response to the ligand, oxygen ligand being oscillated here continuously. So this whole system goes through this oscillation with the net effect of picking up oxygen and delivering oxygen. Here is another simulation that is of interest. If we go into the mountains and the partial pressure of oxygen drops, the oxygen concentration in the blood will respond. It'll drop. And what we are simulating here is the response to the immediate drop in partial pressure of oxygen. And I should note that the DPGase is a very slow first order reaction with a 12-hour time constant or so. And the DPG mutase operates essentially at a constant flux level or as a zeroth order enzyme, zeroth order reaction. So when the partial pressure of oxygen drops, less oxygen will be binding to hemoglobin and more of it will be free of oxygen, giving 2,3-DPG a chance to bind to those oxygen-free hemoglobin molecules, and therefore deoxyhemoglobin will build up. So a part of the 2,3-DPG pool will shift over from the free state to the bound state. And this happens quickly without noticeable changes in the reaction rates. Panel B shows what happens on the longer time scale. You see the blue curve drop very close to the y-axis, which is the short-term response shown on the left in panel A, followed by a recovery of the DPG concentration. When DPG concentration drops, it means that the first order reaction of DPGase becomes slower, and since the DPG mutase flux is a constant, this will lead to a replenishment of the 2,3-DPG pool, and as its concentration rises, so does the flux through DPGase until a steady state is reached again. And the numbers there in the insert in this graph show how the concentration changes at key time points on that curve. So there's a fast response, partial pressure drops, more hemoglobin can bind to 2,3-DPG, metabolism then on a slow time scale rebuilds the DPG level, and the time constant for that change is long. Okay, so that is the process of building a dynamic model for a macromolecule. where the total amount of it is constant, but the ligands can come and go, and you can compute the functional state of that macromolecule. What we will do now is to integrate this module with the glycolytic network or glycolytic simulator built in Chapter 10. That integration is shown here. So part A shows the glycolytic system we developed in Chapter 10. Panel B shows the tight and the relaxed form of hemoglobin. The relaxed form can bind to 4 oxygen molecules that are indicated in the blue color. And if 2,3-DPG binds to hemoglobin, as indicated by the red circle in the middle, it closes off the binding sites for oxygen, and it cannot bind as well to hemoglobin. And we are assuming here that that binding is basically 0. BPG stands for bis phosphoglycerate. It's another abbreviation for 2,3-DPG. This in panel C is the module we just built, and we can now interface that with glycolysis if we can seamlessly integrate these two modules across the dashed line. And panel D shows you the saturation curve again of hemoglobin with oxygen, and reminds you that that shifts left and right depending on the concentration of 2,3-DPG. There is only one integration issue here, and it is shown in this slide. Because the node with 1,3-phosphoglycerate is now a split, so the flux of 2.24 doesn't all go through the phosphoglycerate reaction, but 0.44 flux units, millimoles per hour, go into the shunt. Then the step through that reaction has to be reduced to 1.8 as shown. These fluxes then reconverge at the 3-phosphoglycerate node, and the rest of the flux down glycolysis is the same. So one would have to readjust the perk for the reaction indicated. Of course, the flux through the binding steps, as shown to the far right, are 0. So this is a very, very simple integration issue. We specify the concentration of 2,3-DPG, in this case to be 3.1 millimolar, and the flux of DPG mutase to be 0.44 based on measurements, and then everything else is computed as before. The stoichiometric matrix is shown here. It is a large table, as you can tell, a lot of details. This green portion is what we had before in Chapter 10. This is our new hemoglobin module, and the Rapoport-Leubering Shunt, shown in black here, is what links the two. At the bottom we have pathways, and not going to say much about them, but the first 3 are the same as before, and pathway vector number 4 is identical to the first one, except it travels through the Rapoport-Leubering Shunt, and it has no flux through phosphoglycerate kinase, which is the column that corresponds to a flux load of 0.77, which I guess I rounded up to 0.8 in an earlier slide. What is important about the difference between pathway 1 and 4 is that pathway number 4 produces no ATP, which you can see in the ATP production column being 0 in pathway 4, but being 2 in pathway 1. So glycolysis makes, of course, a net of 2 ATPs, but if the flux goes through the shunt, you make no ATP because the DPGase just splits off the phosphate in the second position, and that never leads to the generation of a high energy phosphate bond. So that's the coupling between the two, and we can now simulate the responses. Here we are showing a simulation to the oscillatory concentration of oxygen in the blood. And if you look at panel A, we see that the DPG mutase is basically a constant. There's a slight wiggling in it because of the downstream ripple effects shown in panel B. I'll discuss in a moment. But the DPGase goes up and down because the concentration of 2,3-DPG oscillates as explained before. Panel B shows the fluxes through the enzymes downstream from 2,3-DPG. So 2,3-DPG goes to 3-phosphoglycerate, and a phosphate is split off there, and then that goes through the mutase, and then through the enolase, and then through a pyruvate kinase. And pyruvate kinase is a slower reaction, and you can see that the rapidly equilibrating reactions of PGM and enolase kind of track the oscillations in the DPGase flux that goes into that 3PG node, and then that oscillation is very much dampened in the pyruvate kinase reaction. Of course, the oscillations in pyruvate kinase will create oscillations in ATP concentration that circles around to upper glycolysis, where glucose is phosphorylated twice, and that's what leads to these slight ripple effects in DPG mutase shown in the blue curve in panel A, but it is very small. So all these oscillations associated with loading and unloading of hemoglobin has a slight ripple effect into lower glycolysis. So there's the input and the output from that node we have now fused with glycolysis, and it has an oscillatory output, and that creates a ripple effect on lower glycolysis as shown. So a few lessons have been learned in this lecture. First, macromolecules can be explicitly represented in dynamic simulators of metabolism and other biological phenomena. Macromolecules have many binding states by low molecular weight ligands. In this case, we considered 2. Binding of these ligands alters protein activity, in this case, the oxygen saturation state of hemoglobin. Subnetworks for a particular protein and all its ligands can be built, where the ligands will be inputs and outputs, and then internal to that subnetwork you have the different binding events to the macromolecule. Ratios can be formulated to represent what fraction of the protein is in a particular functional state, and these ratios can then be used to interpret the use of that macromolecule and the functionality it has. These protein to ligand binding subnetworks can be integrated with the mass action scaffold model of the metabolic network. And this was relatively easy to do in this case, and is relatively easy to do in general. And once that has been done, you can now simulate the dynamics of the low molecular metabolites as well as the binding state of the protein. In this particular lecture, we focused on oxygen binding to hemoglobin. That concludes this lecture, and the next lecture and the final one goes through similar material for a key regulatory enzyme in glycolysis called phosphofructokinase. Many thanks for your attention.